Long-standing mystery solved. Scientists discover new hormone that can double bone mass. This is a breakthrough. In a groundbreaking new study, researchers have identified a hormone named CCN3, which can enhance bone strength, treat osteoporosis, and accelerate fracture healing in breastfeeding women. Experts believe this discovery could revolutionize the treatment of osteoporosis and various fractures across different demographics. Here are the details of this revolutionary finding in the medical world. Researchers from the University of California, San Francisco, UCSF, Parnassus Campus, and the University of California, Davis, UC Davis, have solved a long-standing mystery of how breastfeeding women maintain strong bones despite calcium loss due to milk production. The researchers discovered a hormone called maternal brain hormone, CCN3, in breastfeeding women that helps maintain bone strength despite calcium loss. This hormone is predicted to potentially treat osteoporosis and aid in healing fractures in diverse populations. Results published in the journal Nature resolve the long-standing puzzle of how women's bones remain relatively robust during breastfeeding, even though calcium is drawn from the bones to support milk production. Study author Holly Ingram, a professor of cellular molecular pharmacology at UCSF, highlighted the importance of studying both male and female animals over their lifespans to fully understand biology. She noted that without studying female mice, this significant discovery might have been missed. Osteoporosis and the role of estrogen. Osteoporosis, a condition causing severe bone weakness leading to frequent fractures, affects over 200 million people worldwide. Women are particularly at high risk of osteoporosis after menopause due to a decline in estrogen levels, which normally supports bone formation. During breastfeeding, estrogen levels are also low, yet osteoporosis and bone fractures are much rarer in this period, suggesting that something other than estrogen supports bone growth. Ingram's lab previously discovered that blocking a specific estrogen receptor in neurons of a small brain region in female mice led to significant increases in bone mass. They suspected a circulating hormone was responsible for the super-strong bones but hadn't identified it until now, partly due to delays caused by the global pandemic. In their new study, Ingram and her team conducted an extensive search for this bone-building hormone and identified CCN3 as the responsible factor in mutant females. Initially, the team was surprised by these results because CCN3 didn't fit the typical profile of a hormone secreted by neurons. Upon finding CCN3 in the same brain region of breastfeeding female mice, their doubts were dispelled. Without CCN3 production in these selected neurons, breastfeeding female mice quickly lost bone mass, and their pups began losing weight, confirming the hormone's crucial role in maintaining bone health during lactation. Based on this discovery, they now call CCN3 the maternal brain hormone, MBH. Testing CCN3 and future applications. When strategies to increase circulating CCN3 were applied to young adults, older female, and male mice, their bone mass and strength significantly increased over weeks. In some female mice lacking all estrogen or very elderly, CCN3 could more than double bone mass. Dr. Thomas Ambrosi from UC Davis was astonished by the strength of the bones when tested, noting that sometimes high mineral bones aren't necessarily better and can be more prone to breaking. However, these bones turned out to be much stronger than normal. Ambrosi closely examined the stem cells in the bones responsible for new bone formation and found that these cells were much more likely to create new bone cells when exposed to CCN3. To test the hormone's ability to aid bone healing, researchers created a hydrogel patch that could be applied directly to the fracture site and slowly release CCN3 over 2 weeks. In older mice, bone fractures often don't heal well, but the CCN3 patch promoted new bone formation at the fracture site, contributing to a youthful healing process. Ambrosi expressed excitement about pursuing further applications of CCN3, potentially even in contexts like cartilage regrowth, noting the unprecedented mineralization and healing outcomes achieved with the hormone. A breakthrough in medicine. Many women experience osteoporosis during breastfeeding, which later resolves on its own. Can you briefly explain the connection between breastfeeding and bone loss? To maintain bone health, individuals need sufficient levels of vitamin D and calcium. For women, estrogen, and for men, testosterone levels are crucial for bone resilience. Pregnancy and breastfeeding are risky periods for bone loss due to reduced estrogen levels and increased calcium demand for the baby's development. During breastfeeding, insufficient estrogen levels and increased calcium needs for milk production are significant factors in osteoporosis development. What exactly is the newly discovered hormone CCN3, and how can it help treat bone loss? CCN3 is a hormone found to be secreted by neurons in a part of the female brain and binds to estrogen receptors to maintain healthy bones. It's called the maternal brain hormone, MBH. By blocking estrogen receptors, CCN3 can ensure healthy bone tissue even in pregnant women with low estrogen levels and calcium intake. This hormone's role in fracture healing and maintaining bone health at the fracture site is also significant. How might this discovery lead to new medical innovations? Current knowledge indicates that maintaining bone health requires sex hormones, particularly estrogen in women, and sufficient calcium intake. The absence of osteoporosis despite estrogen and calcium deficiency in experiments is extremely valuable information. This could be groundbreaking in treating postmenopausal osteoporosis, typically blamed on estrogen deficiency. Over 200 million people have bone loss. How prevalent is osteoporosis globally and in our country? It's estimated that over 200 million people globally suffer from bone loss. By 2050, it's expected that 1/3 of women and 1/5 of men over 50 will have fractures due to osteoporosis. What are the causes of osteoporosis? Unchangeable factors include female gender, being over 50, early menopause, and testosterone deficiency in men. Modifiable factors include poor nutrition, low body mass index, insufficient calcium intake, vitamin D deficiency, smoking, alcohol use, a sedentary lifestyle, and certain medications. What are the current treatments for this disease? Osteoporosis treatment is categorized into drug and non-drug therapies. Non-drug therapies include a calcium and vitamin D-rich diet, exercise planning, reducing smoking, alcohol, sugar, and salt intake. Drug therapies are classified as those that decrease and those that increase bone formation. The most commonly used drugs reduce bone formation, with drug choice based on side effects, duration of use, and the patient's fracture risk. Osteoporosis is a significant public health issue due to its insidious nature and the serious fractures it causes, leading to disabilities and even deaths. These fractures have severe implications for families and countries, creating substantial costs. Increasing awareness and educating patients about osteoporosis are crucial since the disease often goes unnoticed until fractures occur. Recently, the use of some drugs that stimulate bone formation is promising for treatment. Monitoring vitamin D levels closely and raising awareness about exercise are also essential. Thanks for watching.